In a previous lesson, we learned about how genetic information is written from DNA onto mRNA. In prokaryotes, that mRNA directly serves as an information source for translation into protein, whereas in eukaryotes, the mRNA is processed in order to allow its export into the cytoplasm, where it's then translated into protein. Now, in previous lessons, we've looked at the central dogma and how information is stored as DNA and then transcribed into mRNA before being translated into protein. So remember that translation involves using the code that has been written onto mRNA from DNA in order to build polypeptides. We're going to be looking at it specifically for prokaryotes, but it's essentially the same in eukaryotes. Here, we have a strand of mRNA. Notice that it's single-stranded and it has the base uracil in place of thymine, as is present in DNA. The mRNA has 3 base sequences called codons, and these codons specify which tRNA can bind to it. In fact, the genetic code is basically written as a sequence of 3 base codons. The main machine for translation is the ribosome, which actually comes in 2 parts, with a smaller part being the 30S subunit and the larger part the 50S subunit. Transfer RNA, or tRNA, are short pieces of RNA that on one side have an anticodon, or a 3-base sequence that is complementary to the mRNA and can therefore bind to it. On the other end of the tRNA is an amino acid. Particular amino acids are associated with particular tRNAs and therefore with particular anticodons.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a 3rd site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the 2nd amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the 2nd transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Now that we know basically how the process works, let's look at a few more details. In prokaryotes, the small ribosomal subunit searches for a particular sequence on the mRNA that's just upstream of what's known as the start codon, and it uses this sequence to know where to bind onto the mRNA. Researchers lined up the sequences of many genes and looked for particular sequences that were common to each of the genes. This picture here shows bases that are particularly common in these sequences. You can see that about 10 bases upstream of the start codon, the sequences don't have much variation. And so we can conclude that these particular sequences are important for some function. In this case, this function is binding of the ribosome to the mRNA. This picture actually shows the coding strand of DNA rather than the RNA. The sequence is the same except that the DNA has thymine instead of uracil. Translation always starts at a start codon, which consists of the bases AUG. You can see that basically all start codons have that same sequence, as shown here by the size of these letters, indicating that there isn't variation. The second thing that needs to happen to initiate translation is that a tRNA needs to bind to the start codon. Because the start codon is the sequence AUG, a tRNA that has a complementary anticodon is needed. The tRNA that has the complementary anticodon always has the amino acid methionine attached to the other end. Once the tRNA has bound, GTP is expended in order to get the large ribosomal subunit to bind to the rest of this initiation complex. The large subunit binds in such a way that the tRNA is located in the P site. In eukaryotes, rather than finding a specific sequence upstream from the start codon on the mRNA, the ribosome binds to the 5' cap and essentially scans down the mRNA until it finds the start codon. Once initiation is complete and the tRNA with the anticodon complementary to the start codon is placed in the P site, the polypeptide gets lengthened. A new tRNA moves into the ribosome and binds to the mRNA. Although many tRNAs may try to bind to the mRNA, only tRNA with an anticodon that's complementary to the mRNA at the A site are able to bind. Hydrolysis of GTP helps make this process more accurate and faster. The polypeptide chain moves from the tRNA at the P site to the tRNA at the A site, and the new amino acid is added to the carboxyl terminus of the growing polypeptide. Remember earlier in the course, you learned about the relationship between amino acids and polypeptides. At one end of a polypeptide is the N terminus, and the other is the C terminus. When polypeptides are made by a ribosome, the N terminus is the first amino acid, and the C terminus is the last to be added to the polypeptide. Once the peptide bond has been formed, the ribosome moves down along the mRNA, and the tRNA from the P site moves into the E site, or exit site. As this happens, another molecule of GTP is hydrolyzed, and the tRNA in the E site is expelled. This opens up the A site, which is then ready to accept another tRNA whose anticodon is complementary to the next codon on the mRNA. When the ribosome encounters a stop codon, UAG, UAA, or UGA, there are no tRNAs with anticodons complementary to these sequences. Therefore, instead of a tRNA moving into the A site, a release factor moves in. The release factor adds a water molecule to the C terminus of the polypeptide, which allows the polypeptide to break free of the tRNA. Finally, GTP is hydrolyzed in order to dissociate the ribosome complex. Okay, we've seen how the ribosome reads 3 bases at a time on the mRNA. These codons are essentially read by the ribosome simply by virtue of having tRNAs whose anticodon complements the codon. Biochemists have figured out which 3 bases on the mRNA correspond to which amino acid being added to the polypeptide, and those are all indicated here. There are a couple of important things to notice. The first is that this table is the code as written on the mRNA. Therefore, all of these three base codons are from the mRNA and not from DNA. The second thing to notice is that the codon AUG is known as the start codon and always results in methionine being the first base at the N-terminus of the polypeptide. Third, there are 3 stop codons that don't specify an amino acid. There are no transfer RNA molecules that have an anticodon complementary to these sequences. Rather, these sequences result in the termination of translation. Fourth, the genetic code is what we call redundant. That is, there are more than one codon for each of the amino acids. In fact, the third base of the codon often doesn't matter, and several different tRNA molecules with different anticodons will be able to bind to the same codon. This flexible base pairing is called wobble and allows all 61 different codons shown here to be able to be recognized by only about 45 different tRNAs in bacteria. Okay, let's look at the structure of the tRNA. Transfer RNA has this general two-dimensional structure with stems and loops binding together regions of the single-stranded RNA. The stems are caused by complementary base pairs being in the right position where loops are folded back on itself. Down at the bottom here is the anticodon, and this is, of course, how the particular tRNA molecule complements a codon on the mRNA. At the 3' end of the RNA is the attachment site for the amino acid. The actual three-dimensional structure of tRNA looks a bit more like this. Now, you might notice that the end of the tRNA, where the amino acid is attached, is very distant from the anticodon. Furthermore, the amino acid attachment site is generic and is the same for all tRNAs. Therefore, an important question is, how does a particular tRNA with a particular anticodon get attached to a particular amino acid? Well, the answer to this is that for each amino acid, there is one enzyme called a tRNA synthetase. This one synthetase is responsible for adding the particular amino acid onto any of the tRNA molecules that carry that amino acid. In this picture, tyrosine tRNA synthetase is the enzyme that's being used to covalently attach the amino acid tyrosine to its transfer RNA. Other amino acids will require different tRNA synthetases, and the specificity of the tRNA synthetase is how each amino acid gets attached to particular tRNAs, even though the attachment site on the tRNA is generic. Lastly, note that this reaction requires the energy of ATP and is sometimes referred to as charging the tRNA, though the word charging here has nothing to do with the electrochemical charges. Okay, we've looked at how a polypeptide is synthesized, but how does the polypeptide get to where it's supposed to go? Some proteins are manufactured by ribosomes on the rough endoplasmic reticulum, and those proteins are destined either for the endomembrane system or for export by the cell. Other proteins are manufactured by ribosomes that remain in the cytosol. As translation begins in a ribosome, the first 20 or so amino acids that emerge from the ribosome are called the signal peptide. This signal peptide is bound to a signal recognition particle, SRP, and if the polypeptide is destined for the rough endoplasmic reticulum, the SRP will bind to a receptor protein on the surface of the endoplasmic reticulum. Once bound, translation can continue, and the polypeptide is formed either in the lumen of the ER or bound to its surface. Polypeptides that are destined for other organelles contain different signal peptides. These signal peptides act as a sort of zip code system so that the protein can be directed to the appropriate place in the cell. One interesting effect of both transcription and translation is that one sequence of DNA can be transcribed into many molecules of mRNA. Each of these molecules of mRNA can then be translated into many polypeptides. Thus, as the cell needs to make protein, it can make a large number of protein molecules rapidly with just a single sequence of DNA. In prokaryotes, translation starts even before transcription is complete. As the RNA polymerase synthesizes mRNA, the mRNA is immediately bound to ribosomes, which start the translation process. Notice here that ribosomes that have just joined the 5' end of the mRNA and are progressing down toward the 3' end only have short polypeptides formed, whereas ribosomes that have been on the mRNA for longer have more complete polypeptides. Almost as soon as the mRNA is completely transcribed, there will be functional proteins. In eukaryotes, however, this isn't possible because the mRNA needs to be processed and exported from the nucleus before any translation can happen.